Hello everybody, this is our third screencast for DNA, and we're picking up right where we left off with our slide for transfer RNA, and we'll get right into the breakdown of the comparison between DNA versus RNA. So, DNA has two strands. It has a deoxyribose sugar and four bases, adenine, guanine, cytosine, and thymine, while RNA has one strand, the ribose sugar, and four bases as well. And those include adenine, guanine, cytosine, and instead of thymine, uracil. So that is our breakdown of DNA versus RNA. And then we'll move into transcription. So transcription is the process of enzymes making a complementary copy of a portion of a DNA strand. This results in the formation of one single-stranded mRNA molecule rather than a double-stranded DNA molecule. And here over on the side we can see our unwound DNA by RNA polymerase unwinds DNA in certain sections, and we make our our messenger RNA strand created based off the DNA. And of course, it's rewound as it goes along as to not damage the DNA. Then mRNA can transport itself out of the nucleus into the cytoplasm to be translated into proteins or amino acids. Full DNA is too bulky to do that. It's too big of a chemical compound to exit the nucleus. So we use messenger RNA to transcribe the information found in DNA, process sent out of the nucleus, into the cytoplasm where ribosomes are located, and the ribosomes translate the mRNA into proteins and polypeptide chains. So the genetic code, we have codons, which we started to talk about today in class. Each is a set of 3 nitrogen bases in mRNA, represents an amino acid. So we have 20 different amino acids and 64 codon combinations possible. There are certain codons which repeat for certain amino acids. Some codons mean stop, and the stop codons are UAA, UAG, or UGA. And then there are also codons that tell the ribosomes to start. So this is how the ribosomal subunit knows where to start from messenger RNA and where to stop from messenger RNA. So this is how, otherwise if it was continuous, the process would go on and on forever, and there would be large, bulky, unusable proteins built. But anticodons, the tRNAs, 3 nitrogenous bases that pair up with mRNA during translation. So start and stop codons are very important in protein synthesis. Without them, it would it would go on uncontrollably and never stop, which would result in some unfortunate folding of proteins, which would be bad. And then we have translation. mRNA to protein. The process of converting the information in a sequence of nitrogenous bases in mRNA into a sequence of amino acids that make up proteins. So this is that process where the start and the stop codons are important. So again, we can see the ribosomal subunit attached to mRNA in the image, and we've got a bunch of free-floating tRNA, which the anticodons, they're bonded to different amino acids, and these amino acids are bonded together, and they are growing a polypeptide chain, and polypeptide chains are large strands of amino acids, and when they get large enough, there's certain chemical forces in play that cause the proteins to fold in certain ways, and that's how different parts of us are built in our cells and in our body, and cells, tissues, organs, all that is built from different folding of proteins. tRNA brings the 20 different amino acids dissolved in the cytoplasm to the ribosome and puts them in sequence. The anticodon on tRNA forms a temporary bond with the codon of the mRNA strand. We have a misspelling there. So, mRNA strand. And then moving on from there, the ribosome slides down the mRNA strand, the mRNA chain, to the next amino acid, the next codon. This forms a chain of amino acids until the codon stops. So until the ribosome will continue to synthesize protein and connect the amino acids chains and build long chain polypeptides until it sees the stop codon. So this is why start and stop codons are so important. They are critical to proper protein synthesis. Otherwise, we would have wild, uncontrollable proteins. So this was a quick video lecture, and we'll talk more about this tomorrow, and we'll pick up mutations over the next few days. Okay? All right. Have a good evening.